Okay, so picture this. A patient is sick. They've got an infection, some microscopic invader that's causing chaos inside their body. For any clinical team, the first job is to basically become a detective. So let's dive in. How do you unmask an invisible assailant, especially when the clock is ticking?
Well, our investigation starts right here at the crime scene. We take a sample from the patient, could be blood, tissue, you name it, and we spread it onto this nutrient-rich plate. The whole goal is to convince these microscopic culprits to show themselves by growing into visible colonies just like these. This is really the first step in the classic algorithm for identifying bacteria.
So today, we're putting on our lab coats, and we are going to follow the clues. We'll trace how the diagnostic algorithm, the step-by-step process that microbiologists use, has evolved over the years, all the way from these painstaking traditional methods to the lightning-fast technology we have now.
We're going to start with the initial mystery. Then we'll get into the classic art of culturing bacteria. From there, we'll see how scientists create what are called biochemical fingerprints, then how the molecular revolution totally changed the game by letting us read the enemy's DNA. And finally, how this entire process got crunched down from days to just a few hours. So let's kick things off with the fundamental challenge. The enemy is completely invisible. How on earth do you identify a suspect you can't even see?
The very first clue usually comes from putting a sample under the microscope. Techniques like the gram stain can tell us right away if we're dealing with gram-positive or gram-negative bacteria, which is a super vital piece of info. But it's kind of like knowing if your suspect has blonde or dark hair. It's a great start, for sure, but it's not nearly enough for a positive ID.
To get that positive ID, we have to turn to the gold standard of classic microbiology, culturing. And this just means giving our bacterial suspects the perfect environment, the perfect food to grow and multiply until we have enough of them to actually study properly.
And this is what that looks like. It's really an art form, and it's called streaking for isolation. A skilled technician has to carefully spread the sample across the plate. The goal here is to thin out the bacteria so much that single individual cells can grow into these pure, isolated colonies. It's a manual, incredibly painstaking process that has been the foundation of microbiology for more than a century. But here's the single biggest problem with culturing: the time. It just takes so long. I mean, sure, some bacteria like E. coli grow pretty much overnight, but a lot of the more dangerous or just plain finicky ones are incredibly slow. As you can see here, Brucella can take more than 3 weeks to even show up. Coxiella, which causes Q fever, can take weeks. And Borrelia, that can take over a month, so long that labs rarely even try to culture it. For the patient, this waiting game can be incredibly dangerous. And on top of that, some of these suspects are just really dangerous to handle. When you hear about BSL-3 safety, we're talking about labs with special ventilation, sealed rooms, full body protective gear, the whole 9 yards. This is what's required for highly infectious agents like Brucella and Coxiella, which can make you seriously ill from just inhaling a few particles. It just adds another layer of difficulty to the whole process.
Okay, so let's say after days or even weeks, we finally have colonies growing on the plate. The next step in the classic algorithm is the interrogation. We run a whole series of tests to build a biochemical fingerprint of our culprit. So this involves challenging the bacteria with a bunch of different chemicals to see how they react. For example, the oxidase test checks for a specific enzyme. The Kligler iron agar test, or KIA, shows if the bacteria can ferment certain sugars. Each test reveals a different trait. And when you combine all these results, you create a unique profile that helps you systematically narrow down exactly what pathogen you're dealing with. Now, this whole process, culturing, then all the biochemical testing, it's slow and it's cumbersome. But then, a revolution happened. We figured out how to stop looking at what the bacteria do and start looking at who they are by reading their genetic code directly. Let's look at a real-world case to see just how powerful this is. It's the summer of 2015, and a deadly outbreak of Legionnaire's disease hits the Bronx in New York City. People are getting really sick, and officials need to find the source, and fast. The big question was, where was this Legionella bacteria coming from? So instead of waiting weeks for cultures to grow, investigators turn to molecular methods. They use a technique called pulsed field gel electrophoresis, or PFG. The easiest way to think of it is as a highly specific DNA fingerprint. It allowed them to take the Legionella bacteria from the patients and compare its DNA pattern to bacteria they found in samples from water cooling towers all over the area. And the results? They were crystal clear. A perfect match. The DNA fingerprint of the strain making people sick was absolutely identical to the one found in a single cooling tower. The investigators had found their smoking gun. And they did it not in weeks, but in days, which let them decontaminate the source and stop the outbreak in its tracks.
This really represents a huge leap forward. You see, the classic algorithm is all based on watching behavior. It takes days or weeks, and it's totally useless for bacteria that you can't even grow in the lab. The molecular algorithm, on the other hand, reads the DNA directly, takes just hours, works on even tiny amounts of DNA, and it's more and more automated. And this molecular revolution hasn't slowed down one bit. It's continued to accelerate, bringing us to a point where the entire diagnostic workflow is almost unrecognizably fast today.
I mean, just look at this comparison. On the left, you've got that classic manual art of streaking a plate. On the right, a modern, fully automated molecular diagnostic system. We've literally gone from a hands-on craft to a push-button science. It's incredible.
And the workflow today totally reflects that simplicity. For a system like this, a technician just injects the patient's sample, let's say from a blood culture that's flagged as positive, into a self-contained pouch, puts that pouch into the machine, and hits start. That's it. All the incredibly complex molecular biology just happens automatically inside that little box.
So what do you get back? Well, in about an hour, the system can identify dozens of the most common causes of bloodstream infections. We're talking bacteria, fungi, the whole lot. And this is the really critical part. It can also detect the presence of key antibiotic resistance genes. This tells the doctor not only who the culprit is, but which weapons might not even work against it.
1 hour. Let's just let that sink in for a second. The entire identification algorithm, which used to take days of waiting and culturing and testing, is now compressed into a single hour. And this is why it all matters so much. For a patient with sepsis, every single hour of delay in getting the right antibiotic dramatically increases the risk of death. Faster answers mean doctors can prescribe the correct, targeted therapy so much sooner. It saves lives, it reduces hospital stays, and it helps us stop outbreaks before they can get out of control. So this brings us to a final thought. We've managed to shrink the diagnostic timeline from weeks down to a single hour. The algorithm for identifying our enemies is faster and more precise than it has ever been. So, the question now is, what's next? With identification pretty much solved, what becomes the next great battlefield in our ongoing war against infectious diseases?